So anyway, I'm very, very excited to introduce our featured speaker for tonight. Christian Schwarz studied ecology and evolution at UC Santa Cruz, where his interest in the world of fungi became irrevocable. Their seemingly endless forms, from the grotesque to the bizarre to the sublime, I totally agree, feed his curiosity. Christian now teaches natural history of fungi and community science through the Norris Center at UCSC, and he is co-author of the gorgeous book, doesn't say that in his bio, but I'm editorializing here, Mushrooms of the Redwood Coast, which is just, a stunning, stunning book. He's an incredible photographer. So definitely check that out, Elizabeth. I want my copy of that back someday. I'm just kidding. You might have already given it back to me. I can't even remember. But anyway, and he also has served on the IUCN Red List Working Group for North American fungi, which is really, really cool. That's how fungi get listed as being, threatened or near threatened or vulnerable or not vulnerable. But anyway, so Christian spends his time studying the ecology and natural history of California, Baja California, and Arizona. And he's passionate about biodiversity in general, and especially the philosophy and practice of community science. So I was saying to Christian earlier that I saw him speak, maybe like a year ago, and I was really so taken with his talk, which I think will overlap somewhat with his talk tonight. And I've been really wanting him to join us ever since then. So very warm welcome to Christian, and thank you so much for being here. Very excited.
Sure thing. Thanks for having me, Annie, and nice to see you all. I know some of you in person, but I think relatively few. I saw Jay Justice's name earlier. Good to see you, sort of, Jay. I wish I was out there with you guys. I see Adolf Cheska. Nice to see you, Adolf. And for those of you who I haven't met, don't know, I'm hoping to actually get out back to the East Coast like I did 2 summers ago, maybe next year. Oh, Alfredo Justo is here. Hi, Alfredo. There's a lot of folks now, so I'm not going to scroll through everyone, but if I've met you before, hello. And if I haven't, welcome to this talk. I am from California. So my bias in terms of mushrooms that I know is Western and Californian. What I call the Pacific states, any state that has a shoreline on the Pacific Ocean. So I know relatively little about Alaska. I've been there once, and I talked to Noah, my co-author with Mushrooms of the Redwood Coast. He studies Alaskan mushrooms a lot. But Washington, Oregon, and especially California are my home range. And that, like Annie mentioned, I've spent some time in Arizona in the past 10 years. And then whenever I am lucky enough, I go down to Baja California. But unfortunately, I haven't ever really intersected Baja at the same time that it's having a really excellent mushroom season, although that's happening right now and I'm not there. So what am I doing? I don't know. I should go right now. But the point of me giving a rundown of where my geographic interests lie is that's what tonight's talk is about. It's about the geography of species distributions of macrofungi, mushrooms. So I'll just turn my screen sharing on right now and we'll get into it. Okay, almost. All right, can everyone see that? Okay, here we go. And there are kind of a lot of people in the group tonight, but Annie, if you're watching...
Yeah, I can watch the chat for you if you'd like.
Studios. This week on the New Yorker Radio Hour, an unassuming Supreme Court justice who made an outsized impact on the Supreme Court.
Is that me?
That's the New Yorker Radio
Hour. I don't know. Don't think it's me. I don't know who that is.
Today, I want to play.
Okay.
That's weird. But anyway, I can watch the chat for you if that would be helpful.
Yeah, if you feel like there's a question or something that needs to be addressed, sort of it makes more sense to do it in the flow of the talk, I'm happy to take questions right in the middle. And then obviously, like usual, we'll just do a bunch at the end.
Perfect. Sounds good.
Okay, cool. Here we go. Am I in presenter view? or not. I can't tell what you guys can see. Okay, there we go. All right, so the talk is called Bioregions of Macrofungi in the United States, and I'm going to walk through what I mean by all that in a second here. But I think what is clear to everyone from the start is that when I say a bioregion for macrofungus, what I mean is where that thing lives, where that species lives on a map, spatially. But that's not all that I mean. Can anyone guess which macrofungus has this distribution globally? This is one where you can just unmute yourself, or I don't know if there's guesses coming into the chat, you can read them, Annie.
Amanita muscaria.
Any other guesses? I saw someone said split gill.
Steps. You
What
got
was
a
that?
turkey tail.
Turkey tail.
Russulas.
As a genus, perhaps. Yeah, I'm talking about an individual species, though. So split gill is an excellent guess, but I actually suspect if we had a map of split gills, it would be even more fully red than this. Jack Johnson got it. This is Amanita muscaria. And this is not a map. This map wouldn't have looked like this, say, 1,000 years ago. If you were to map where Amanita muscaria lived in the world 1,000 years ago, many of these points would be missing. Notably, for example, New Zealand. Amanita muscaria is a anthropogenically introduced species into New Zealand, where it has started to spread out of Monterey pine plantations and Douglas fir plantations into native forest. So the point I'm trying to make with this map is that this is a fungus that has made it. By whatever account you want to judge it, this mushroom has been extremely successful in either dispersing by itself or being dispersed by other organisms, most notably human beings around the world. So it's now got basically 6 continental reach. It's everywhere to some degree, usually growing with imported trees in places where it's not native, but sometimes it has made the jump to native habitats there. Okay, but let's contrast that to something like Lepiota luteophila. This mushroom is only known from one stand of trees basically anywhere in the world. Oh, you can't really see that background there. I'm sorry. Hold on one second. Well, I'm just going to remove this real quickly and then go back into. It is only known from one stand of cypress trees in coastal California. It probably had a much larger distribution, and it may even still have a much larger distribution, or at least somewhat larger distribution, but we don't know that yet. And to contrast these two mushrooms tells us that there is something different about their biology. They do different things with their physiology, with their biochemistry, with their spore morphology, with their associations with the nearby plants. They are not the same. And there's a gradient all the way from Amanita muscaria on one end of the spectrum to something like Lepiota luteophila on the other end of the spectrum. We don't typically see things being so successful and widespread as Amanita muscaria, but at the same time, we don't typically see things being as rare and restricted as Lepiota luteophila. Most mushrooms fall somewhere between these two extremes. And there's a story to be told, or really multiple stories to be told, about what shapes the distribution of any given mushroom. It's a very rich thing to think about. If you start to sort of mull over why a certain distribution looks the way it does, it leads you down roads of questions that are cultural. What do humans do that shapes that? What has geologic history done? What has deep time climate history done? What has vegetation turnover, sort of floristic turnover in a human lifetime even, how has that influenced what mushrooms we see in a particular area? And all of these processes operate at different timescales. So here's an example of a mushroom that some of you are probably very familiar with. The green gill, Chlorophyllum molybdites, is extraordinarily common in California, and it's actually common across much of the United States. But this is a mushroom that we don't have as much historical data about it as I'd like, but if you kind of take a little fractional view of what you can see on iNaturalist just in the few years that it existed, the platform has existed, that is, you can see that even between the 5 years between 2018 and 2023, there has been a slight northward creep and overall general expansion of where this species has been recorded. And that northward creep has something to do with anthropogenic climate change. There are warmer nights and more periods of uninterrupted warm nights, even at northern latitudes and even in coastal areas like my home county of Santa Cruz. We didn't have any records of Chlorophyllum molybdites until 1 or 2 years ago, when we finally got our first one. Because mostly the coastal influence is enough to keep fog rolling in and cool evenings, and this mushroom can't quite get established, but we're starting to see it creep up into areas where it historically hadn't been. I mean, you've got records all the way up into Manitoba and British Columbia now, so this thing is clearly on the move. And it raises the question, at least, of where is it native to? Where was it originally found before? I mean, I wouldn't actually say that it's not native in all these other areas, but I would say that it is tied to an anthropogenic shift in climate. So it's not that it's not native, it's a species that's opportunistically expanding under this sort of regime change in climate that our carbon emissions have caused. But what I was going to ask is, where was it before that warming trend spiked, before this major shift in climate change? I think I have seen it, well, I know that I've seen it in rather sort of native looking, non-anthropogenically influenced, non-grassy, non-watered areas in the desert in New Mexico and Arizona. And I wonder if it was just something that sort of subtropical desert fringes, and it's expanded northward and really taken advantage of urban areas, urban planted lawns and watered areas. But that's a question for another time. Okay, those are 3 examples of the kinds of stories that you can begin to tell to explain a distribution. But other factors that you need to be aware of in thinking about mushroom distributions are things like what is represented on this map. The answer is already on the screen, but if you hadn't already guessed it, this is a map of elevation across the United States. So you can see that it really is true that the Western United States has a monopoly on high elevation. If there are mushrooms that are somehow tied to this, what we call abiotic factor, which is to say it is a factor not related to the living ecology of the place, but to something sort of about the physical nature of the place. Now, I should also mention that those 2 things never come separately. They always come tangled up in complicated ways. But rainfall, soil minerals, elevation, hours of daylight, hours of dark, those are the kinds of things that are abiotic factors that might shape where species live, as opposed to things like biotic factors, where a certain plant host is distributed, or where a pathogen is distributed, or where some sort of natural competitor of a mushroom is distributed. Those are the things that we might call biotic factors that shape or influence the distribution of a species. So you can see that there's really clear patterns of elevation across the United States that influence where mushrooms might live or where any other organism might live. But the same goes for average annual precipitation. You can see that there's a real difference between a place like North Carolina. Western North Carolina has extremely wet areas, as does the Olympic Peninsula up in Washington, as opposed to something like the lower Sonoran Desert or the Mojave Desert that you get in southeastern California or southwestern Arizona. There's also this almost perfect band running from North Dakota to the tip of Texas that splits the country in 2. And weirdly, that corresponds pretty well with that last image I showed you about elevation. It correlates with this elevational band that splits the country in 2. And you'll see that there are some mushrooms that, quote unquote, obey this division of Eastern and Western North America to a really spectacular degree of precision, even though there's no, border checkpoint where they can't cross. There's just something about the confluence of biotic and abiotic variables that prevents them from moving to the west or to the east of that line. Okay, so now what I want to think about is the biotic variables in sort of aggregate. We can divide vegetation assemblages. If any of you have spent, a lot of time traveling between zones in the United States, you'll know that the prairies of the central United States feel really different from the Great Basin, and they feel really different from the southeast, when you're in the deep, humid swamps of Atchafalaya Basin in Louisiana, for example. And they're also really different from what you would experience if you went up to Alaska. And that's because there are these sort of co-occurring assemblages of plants that give places kind of stable, always fluid. There's always exceptions and sort of blurry boundaries, but you can begin to identify or characterize different regions at varying degrees of coarseness, varying degrees of scale. So over on the left, I've got a very, very coarse division of the United States and Canada by these vegetation types that are, called Boreal, Arctic, Cascadian, Laurentian, Prairies, Dixon. I don't know whose divisions those are. They're not my preferred ones, but they can give you a sense of how some people have approached this challenge of characterizing where things live. And over on the right, you can see the opposite approach, where it sort of divides the country, or these two countries, much more finely. There's, upwards of 100 divisions, much more sort of specifically parsing these vegetation assemblages based on someone's rubric of what they thought was an important distinguishing feature. If you look in Florida, there's these little purple streaks. Someone thought those were distinctive enough to merit their own division. So I am trying to aim in this talk tonight somewhere between these two extremes of very, very coarse over on the left, almost the size of the whole country in the case of the boreal forest, and very, very fine over on the right, because we don't really have enough data about mushrooms to do the kind of fine division you see on the right, but we have enough data to start making finer divisions than you see on the left. So I'm aiming for an in-between here when I ask the following question, which is, what are the major mushroom bioregions of the United States? And my talk tonight really is more of a question than an answer. I have some proposals for what some major mushroom bioregions are, but I'm not committed to them. I don't think these are necessarily true units. I don't think I've characterized them precisely enough to even be super useful to myself. But I think some of them have a good argument for why they might merit division as a discrete unit of a mushroom bioregion. But I'm asking this question because I want you to ask yourself when you're going in your travels, not only locally, but across the United States, to start thinking about this question when you make observations of species, to ask yourself about where the limits are, where the boundaries are of common species. That's one of the things I like when I focus on biogeography, is that the common species take on an increased importance. A mushroom that you take for granted as being everywhere is never actually everywhere. There are always interesting gaps and holes and funny sort of finely divided edges to where a species lives that can teach you something if you remind yourself to pay attention to where a mushroom isn't. And that is a much harder thing to be aware of because it's sort of negative data. A mushroom that's not there can't be seen. But over time, by adding your observations to iNaturalist, you give people like me the opportunity to go probe these questions by looking at a map and staring and thinking hard about it and testing some hypotheses in sort of oblique ways that we can do if we have enough data. So there really is no scale too small to focus on. In your county, even, you can do this, but especially if you travel, pay attention to where mushrooms are and aren't. And over time, these sort of network of mushroom clubs across the country will be able to sort of help each other inform an answer to this question. What are the major mushroom bioregions of the United States? So I'm going to walk you through some of how I think about this country in terms of its mushroom diversity. Okay, California. This is where I'm from. And I think that there are two main things that give California its flavor from a macrofungal perspective in terms of the diversity and uniqueness. Number one are the redwoods. So the redwood bioregion extends from about central California all the way to the Oregon border and just slightly over the Oregon border. And it contains a remarkable diversity of very unique species that you don't find in other kinds of habitats, including things like what you see on the left here, waxycaps and entelomas, these blue, yellow, purple, pink mushrooms, very brightly colored, as well as this sort of microglossum up here on the right, this green earth tongue. But we also get some other things growing with, for example, our live oak flora, our live oak assemblage. These are mycorrhizal, unlike the redwoods, or ectomycorrhizal, unlike the redwoods. So they get a lot of Cortinarius and a lot of boletes. And some of these are quite distinctive, that they don't really extend outside of the Californian bioregion. Because if we go back here a few slides, you'll see virtually everyone who ever splits up the country into bioregions acknowledges that California is its own region for a very particular set of reasons. One is that to the north, it gets cold. To the west, it gets very wet. That's where the Pacific Ocean is. To the east, you get Great Basin and deserts sort of forming this barrier to dispersal of a lot of organisms. And then to the south, it's also very hot, and you get the finely divided Baja California Peninsula and northern Mexican deserts. So California really is a biogeographic island in a very real sense. And the Sierra Nevada and Cascades serve to further strengthen that eastern-western division along the eastern boundary of California, separating it from the rest of the contiguous 48. So preventing the dispersal of organisms, not just mushrooms, plants, animals, herps, all sorts of things can't make it over those mountains into the western part of California. So California has this sort of redwood and live oak overall flavor. We also are the site of some of the world's highest conifer diversity. So if you go up into the Klamath Mountains of northwestern California, you get 14 or 15 species of conifers in a very small area, a single square mile. So it really, and obviously those conifers bring the mushrooms associated with them. So you can get very distinctive assemblages of fungi just based on these few trees that exert an undue or sort of an exacerbated influence on the fungi that might be found in a particular area. Another special thing that's not just particular to California, but which does give California a very special flavor. This also happens in Washington and Oregon, as well as you'll see, just some of the other Western states, but what I call the Niveomycota, the snow mushrooms. And really the flagship mushroom, or the most iconic mushroom of the Niveomycota, is the snow rose waxcap Hygrophorus getzii. This is a mushroom that grows through the melting edges of snowbanks and sort of melts its own hole through that snow. It's a spectacular mushroom, a really amazing thing to see. And it is a mushroom that has a very concrete set of both biotic and abiotic variables that shape where it grows. The reason that you only find this mushroom in these places is that it needs snow and it needs mountain hemlock. Those are the two variables that overall explain 90% of the distribution of where this mushroom is. Now, there are places with mountain hemlock and snow that don't have this mushroom, and then you get that remaining 10% of variation in distribution that you need to explain. But if you put those two variables together, you can basically come up with an accurate map of where this mushroom lives. So it's very widely distributed through the Cascades of Oregon, as well as in Northern California, all the way down to Sequoia. Well, no, not as far as Sequoia, but anywhere where there is mountain hemlock, you have a chance of finding this mushroom, because mountain hemlock itself pretty much correlates with places where there's snow. This record from Idaho appears to be valid. It's legit. And I wonder about how much more widely dispersed or widely distributed it is in the sort of interior equivalent of the Pacific Northwest that goes up into the panhandle of Idaho and into the sort of Canadian Rockies. So there's probably more dots on the map to fill in for this species, but it is very concrete. If you want to go find this mushroom, you know where to go looking. There are many places you can rule out right off the bat, and relatively few places left over that even have potential for its distribution. But there are other members of this snowbank flora. I'm going to go clockwise here, starting at the upper left. This is Donadinia nigrella, this beautiful black cup fungus that grows out of the edges of snowbanks. Nevada Gastrium nubigenum in the middle there. That thing smells like wintergreen. It's an amazing Secotioid foliota. Number of cup fungi like this Lachnella on the upper right. A couple of mycenas down in the bottom right and middle. And then Crepidotus that grows on branches under the melting snowbank. So it really is a pretty diverse set of more than a couple dozen, probably dozens of species. I don't know if anyone has actually put together an exhaustive list of all the snowbank fungi, but that would be something very much worth doing, because they're all of interest from a conservation perspective because snow regimes are changing very rapidly in the Western United States. And the Mycota, this sort of mountain snowmelt phenomenon of fungi fruiting when the snow melts in the spring, is not something that happens everywhere in the world, strangely. There are mountains with snow on every continent, and yet it appears that something about the Western United States is special in terms of fungi that have evolved to take advantage of that pulse of moisture in the spring. And like I said, climate change is pretty dramatically impacting how quickly and how deeply the snow accumulates, as well as how fast it melts. And so any fungus that's dependent on that pulse of moisture is going to have to evolve quickly to change or to keep up with that change, or else potentially risk having a much narrowed or even absent reproductive season. Okay, moving north from California, following the Cascades, those mountains that I just mentioned, we get into the Pacific Northwest. So Oregon, Washington, and British Columbia. This is an area given a great deal of its character by these volcanoes and coniferous forests. And so you get mushrooms like Phaeocolybia, Ramaria, Cortinarius, Amanita, and then in the middle, at the bottom there, this photo by Connor Dooley of Squamanita. Some of you are probably familiar with Squamanita fimbriata, Squamanita umbonata from your area. I've never seen any Squamanita of any kind. These are extraordinarily rare mushrooms wherever they occur, but this is a mushroom that we didn't really know existed in this one that Connor photographed in the Western states until very recently. And it's something that definitely gives the area its flavor. These sort of rare species are oftentimes good indicators of a bioregion, but not always. Sometimes rare species are surprisingly widely distributed. And that brings me to another sort of point I want to make about thinking about bioregions, is that you should try and look for an emblematic or iconic species. When you come up with a proposed bioregion in your head, you should say, okay, I've got this idea of an area that seems to have a distinctive character or distinctive flavor, but what is a mushroom that fully inhabits that region and really seems to represent it? And I'll show you some examples of what this looks like. One that I always think of for the Pacific Northwest is these Polyozelis, which recently was split up. Well, not even recent anymore, but for those of you who don't know, Polyozelis multiplex is not the only Polyozelis. There's 5 or 6 species now, and they all have slightly different distributions. But if you go to the Pacific Northwest, Polyozelis is something that you can expect to see if you get into a nice old growth conifer forest. But perhaps an even better rare species that sort of inhabits the Pacific Northwest bioregion is this noble polypore, Brygioporus nobilissimus. It's one of the few macrofungi that has attracted serious conservation attention from land managers at the state level because it's so rare. For those of you who've never heard of it, this is one of the largest fruit bodies of any macrofungus in North America. It can be 3-4 feet wide and 2 or 3 feet deep. A really enormous fruit body, deep sort of shag carpet like upper surface, and grows on the base of old growth grand fir, so a true fir. And there's relatively few old growth grand firs left because they get logged. They're nice timber trees, and so a lot of them have been removed from the forest that they exist in. And curiously, although the mycelium of this species has been found distributed pretty widely in grand fir trees, the fruit bodies appear to be really rare. So it doesn't appear to form sexual reproductive fruit bodies very often, even though it might be more widely distributed throughout the Pacific Northwest than the records might suggest. But it does seem to inhabit the area that I talk about when I talk about the Pacific Northwest. It anchors the sort of boundaries of what that region looks like in my head. Okay, so moving north from California, then through Cascadia, north still, the last of the Pacific states. This is Alaska, obviously. And the way I think about Alaska is that it sort of connects the North American landmass to Eurasia by virtue of sort of the Aleutian Islands. There's actually very, very little distance between Alaska and far eastern Russia. And so some of the spores of fungi I guess, have a chance of dispersing little by little from one landmass to another and dispersing down the west coast of the United States. So it's sort of potentially where we get some of the propagules of Eurasian fungi setting up sort of shop in the North American landmass and potentially spreading outwards from there. And obviously in deep time, Alaska wasn't always what it looks like now. There was periods of more glaciation and less glaciation, more extensive forest, less extensive forest. So that dispersal ability may have made a much greater difference in deep historical time or prehistoric time. So this is the edge of a glacier on the Seward Peninsula, and it doesn't look like there's much there, but you can see these little smears of vegetation, little green islands of plants living on these pumice fields, basically. And that yellow mushroom at the upper right there was growing out at the edge of this glacier. So in places that are very forbidding, mushrooms find a way to make an existence. But here are some mushrooms that you find in Alaska that are sort of quite distinctive. They have sort of a different looking group of Amanita muscaria that are a little bit more similar looking to the Eurasian version of Amanita muscaria, not so much like, although they are currently considered the same species as the one that we get in California, they look slightly different. They have differently colored universal veil material. They have a slightly different stature and different range of hosts that they associate with there. Cystotrema confluens is the one on the upper left there. Down at the bottom left, that gray russula is something like Russula consobrina. It's not something you really expect to see anywhere else in the Pacific states, at least as far as I know. Over on the bottom right there, Plicateuropsis crispa is something that you're all really familiar with, but that's a mushroom that really is not very common on the Pacific coast, but in Alaska, it's everywhere because there's lots of birch trees. So it's more like what you guys have back east. And then in the middle, at the bottom there, that's Hypocreopsis. That's an amazing mushroom. In Finland, they call it troll hand. And it's really a mushroom you don't expect to see further south. It loves those northern birches. So Alaska really is a bioregion all on its own. No matter what way you slice it, there's going to be some sort of Alaskan bioregion for American macrofungi. Here's an example of a bioregion that is not so easily geographically circumscribed. It's more outlined by a genus, not even one species, but the whole genus of larch, Larix. These are conifers that are famous for changing color in the fall, turning brilliant yellow and losing their needles, unlike most other quote-unquote evergreen trees. And they host this very particular diversity of swillis, slippery jacks. And these are not the only three. There are a number of different slippery jacks, as well as some waxycaps that are larch specialists. And if you know anything about swillis, you know that they are hyper specialists. They oftentimes only grow with one or two host trees and nothing else. So if it wasn't for the larches, we would be missing this whole assemblage of fungi. But larches, it turns out, connect the far northwest of the United States with the far northeast of the United States by traveling across what we call the boreal forest of Canada at the upper prairies. And so they act as what I think of as kind of connective tissue, where they bridge the east and western halves of the United States, and they allow fungi to disperse more or less uninterrupted. You'll find some of the same larch associates in Maine that you do in Washington. And that's true of the boreal forest in general. It's not just larches, it's all sorts of coniferous trees that grow in, very dense forest all the way across the northern tier of states and northern Canada. And when I was thinking of what a flagship mushroom might look like for this area, I came up with Sarcosoma globosum. It's a rare species, it's associated with old growth, and it really doesn't fully inhabit the whole boreal zone, but it is quite emblematic of it. It's an amazing mushroom, can be the size of, a grapefruit. Witch's cauldron is one of the evocative common names for it. I've never seen it, I would love to, but this is an example of something that might be a good iconic mushroom for the boreal forest. Now descending down from the boreal forest, following the Canadian Rockies down into the center of the country and all the way down really to northern Arizona, you get aspen stands, you get conifers, you get very rugged terrain, you get lots of snow in the winter and monsoonal summer thunderstorms, and you get things like Lexinum fruiting in abundance in that aspen forest, Matsutake fruiting with the conifers, obviously the very famous Boletus rubriceps, the red-capped porcini, ruby-capped porcini. It's a different race, so to speak, of the edulis that we get in the Western United States, like in California, we don't get rubriceps. Some people think there's not enough genetic divergence in the two to call them a separate species, but if you ever go to Colorado or Arizona, you know that they look quite different. They're much darker red. Alloclaveria purpurea, this purple spindle coral down at the bottom right, is an interesting one because it's all over the place in the Rockies, and you guys probably see it in the Northeast, but it's quite rare in California. You only get it in a little smidgen of Northwestern California. Coastally, you get it in Washington and Oregon and the dunes, but it's got an interesting distribution, and I'm not really sure whether this represents one species that's got a sort of disjunction across the middle of the continent, or if it's 2 or 3 different taxa that are just similar. They've got this distinctive purple morphology. And what I like about examining these maps is sometimes they give you clues about where to go sniffing if you want to look for species that are actually different but currently being considered under one name. Disjunct sort of blobs where you get species ranges that are highly separated are always a good clue that something more interesting might be going on, and you should not take for granted that the species is the same all the way across its range. This might be your first indication that something more interesting is going on. But the opposite can also be true. The opposite can be true because there are mushrooms that once had a continuous range that was then disrupted or split by some sort of set of geographic barriers arising, like a mountain range or a desert sort of becoming more arid over geologic time, or humans, deforesting an area and creating sort of a artificial gap in the distribution of a species. But if you follow the Rockies further south still, you end up in a different mountain range, not the Rockies. They come up and they touch the northern sky islands of New Mexico and southeast Arizona, which are actually better considered as extensions of Mexican mountains, the Sierra Madre Oriental and Occidental, the eastern and western Sierra Madre. These are mountains that are, I think of them as reaching north from Mexico and barely crossing the border. This is an area where you get all sorts of influence of tropical and subtropical species. If you go to the Chiricahua Mountains, which are pictured here, looking from the north into Cave Creek, this is an area where you have 5 different species of big cat. You have mountain lions, you have bobcats, you have coati, sorry, not coati, jaguarundi. Historically, there was jaguar, and I'm forgetting one, ocelot. But this is an incredible diversity of big mammals. There's javelina, things that you just really don't expect to see. Elsewhere in the United States, coatimundi are a possum family, a raccoon family member that is sort of very typical of the subtropical forests further south in Mexico. But they bring in fungi and plants with those plants that are hard to find anywhere else in the United States. So you get a bunch of different things going on the Sky Islands. Not only do you have sort of endemic species of the Sky Islands, but you also get these remnant eastern boletes and eastern tricholomas and eastern mycorrhizal species, especially associated with oaks, things like the strobilomyces in the top center of this image. I like this bottom center image, but you get mushrooms fruiting right next to cactuses, which, oftentimes feels like a very, almost like an oxymoron. Cactuses being so evocative of dry climates and mushrooms being so evocative of rainfall. But that's what you get in the Sky Islands, is you get these brief bursts of monsoonal moisture, making a rather arid landscape very lush and very rich with mushrooms for a very small period of the year. But you'll recognize, folks from DC will recognize Boletus frostii and strobilomyces, and maybe this Omphallotus subaludens, as well as Russell's, is that Russell's bolete?
Yeah, Areoboletus russellii, yeah.
I don't know if you have another common name for it, but Russellia, whatever you
That's
call
what we
it,
have. Yeah.
is also present, although very rare in the Sky Islands. And what do these indicate? They are clues to a deep time story. They're clues that there was once a connection between the eastern oak forests and the remnant oak forests of Arizona. So you can think of a past era where the climate was a little bit wetter and forest was more contiguous all the way across the eastern United States and the Gulf Coast west into New Mexico and Arizona, and connecting the forests there with the forests of northern Mexico, and forming sort of just a continuous belt of green. And that potentially these mushrooms dispersed all through that. And then as climate changed and became more arid and cooler, that this is obviously before human time still, because we warmed it significantly recently, that those forests became more disjunct, that there were more gaps in the distribution of oaks, and that what we see in Arizona now are remnants of that ancient connectivity. And these are really sort of barely hanging on. They're remnants of a past climate that still have little footholds in these various high mountain ranges, where there's still enough moisture and there's still oaks hanging onto these canyons where these mushrooms can make a living. But they're generally rare. Like, you're not going to go out to any spot in Arizona and find frosty eye. That's a special thing there. And anytime you see it, it is an indication that you're in a place that at one point had some sort of linkage to a much broader bioregion that we no longer see being connected. I've tried to piece together sort of the story of oak distribution across the United States in deep time, but it's a complicated story that I still haven't quite gotten my head around. I haven't quite gotten temporal milestones for when that connectivity was at its maximum and when it became interrupted. So it's still something I'm hypothesizing and researching about, but it's not a perfectly neat story yet. It's something I need to continue to think about. But another thing to remember here is that also Mexican oak species disperse northwards into the United States along these Sky Island routes. So we get Mexican clades of oaks that aren't represented elsewhere in the United States that have their own assemblages of fungi with them. Quercus hypoleucoides, white leaf oak, is an example. If you go to a white leaf oak forest in the Sky Islands, it's got all sorts of cool mushrooms that grow with it. Here's a mushroom that some of you are probably familiar with, and I just want to point out how remarkably it obeys the east-west division of the United States. It's almost like a straight line through the Dakotas to the tip of Texas, and this mushroom seems to say, I shall go no further. This is my boundary. Even though there's plenty of oaks for it to grow on in the Western United States, its preferred host genus, it somehow either doesn't or can't disperse across this boundary. Okay, skipping to the northeast now, the land of maples and birches and fall colors. You can see this picture showing these dramatic deciduous forest colors. This is an area that is very rich in macrofungi. For those of you who haven't gotten into the far northeast, you get hemlock-associated species, you get birch-associated species. In fact, I think one of your members was talking about this Lactaria subpurpurascens earlier tonight. Tectella, up in the upper right there. Tricholoma, this one was recently described down at the bottom left here. Annie, I don't know if you remember the new name for this. I've forgotten. It's the one with all sorts of black scales on the stipe. I took a picture of it before it was described, and I have forgotten to update my notes, so I can't remember what the new name for this is. It's like Atra something, I think. I don't know. And then Leucophiliota decorosa in the upper left there is an example of a potential flagship mushroom for this area. So if you look at its distribution, it does seem to inhabit the Northeast more than, say, the mid-Atlantic states. It's not really present in the deep South. But if you look up in British Columbia, there is this sort of uncomfortable dot up there. There's an uncomfortable dot in Mississippi that appear to be legitimate records of it. So this shows you that these bioregions are always blurry. They're not perfectly circumscribed. They're not neatly edged. These boundaries always have some amount of uncertainty to them. Now, that's not true for mushrooms that are highly tied to a single host tree or some other sort of highly restricted abiotic factor. So compare that, for example, to something like Rhodotus. This is not highly tied to a single substrate, but you can see that although they are quite similar, they're not the same. They don't inhabit exactly the same region of the Northeast, but they are probably largely co-occurring. This Rhodotus probably occurs a little bit further west on average, whereas Leucophiliota extends further into the Northeast, but they overlap, and they might signal either somewhat subtly different bioregions, or maybe you could consider them part of the same bioregion. Maybe they're close enough. But they don't, for example, they're not equivalent to something like Gyroporis cyanescens, which is really, really more northern, with some outposts in the Appalachians, and what I am pretty sure is probably a disjunct species that's not the same in Florida. And then you get this uncomfortable dot in Arizona. And that uncomfortable dot probably represents, and that's a legit find. I've actually been to the exact spot where it was found. I didn't find it myself, but I went to the spot looking for it. It's the only record from the Western United States. And it's a clue. It is a holdover from when that eastern hardwood forest extended further across Texas and New Mexico and down into the Sierra Madre Occidental. So these records do show up, and they do raise questions about what the bioregion should be circumscribed as now, and really what our goals are. That should always be in the back of your mind. What is even the point of establishing a bioregion? It can help you talk about where conservation might focus its efforts. Are there bioregions that include a lot of threatened taxa that all sort of co-occur? That's one way to think about what a bioregion might be useful for. But a lot of it, to be honest, from my perspective, is simply interest. I'm simply interested in understanding where these co-occurring sort of assemblages of fungal diversity are. Here's one that is very clearly its own bioregion that is not neatly circumscribed or overlapping with other species. It's got its own deal going on. It's not a central state mushroom because it doesn't go very far south. It's not exactly a northeastern mushroom because there's whole big chunks of the Northeast that it's not in. It's not a southern mushroom. It's not really a plains mushroom. It's kind of a Great Lakes mushroom, but it extends a little ways away from the Great Lakes. I think Great Lakes are going to end up having their own region sort of associated with them. But this amazing mushroom that I've never seen, I would love to see, it's totally on my bucket list, Paragyrodon sphaerosporus, has its own bioregion that probably also indicates where we should go looking for some other interesting mushrooms that have similar distributions. Atlantic white cedar is a very widely distributed plant, but it contains an assemblage of mushrooms that are actually somewhat similar to what we get with red cedar and yellow cedar and coast redwood in the Western United States. You get waxycaps, you get entalomas, you get a lot of what we are now starting to call the chegg assemblage. So clavarias, hygrophorus, entalomataceae, and the geoglossaceae, the earth tongues. They are often co-occurring, as well as some other mushrooms that are not part of the core chegg assemblage, but they are often found around them. But what I want to point out about Atlantic white cedars is that they had the misfortune of being useful to people. They were excellent lumber trees, excellent timber. They don't resistant to rot, and even the boggy sort of swampy substrate that they grow out of is sometimes so rich in iron that for a time people were boiling the water to make swamp iron because it was so rich in iron you could get these deposits of iron left over. Apparently, I didn't know this until very recently. So even after logging all the trees out, they then went after the bog itself. So some of these were very deeply impacted, and so you don't get a lot of remaining stands of old growth Atlantic white cedar, which is a shame because it's clearly a distinctive bioregion for fungi, extending, all across the eastern seaboard, but it's the only place to find. So here's sort of the distribution of Atlantic white cedar, broadly speaking. But it's also the only place to find Bellew's cedar tooth. This is one of the rarest mushrooms in North America. It only grows on Atlantic white cedar, and as far as I know, only old growth Atlantic white cedar, and there's relatively little of that left. I would love to see this mushroom. There's only one Echinodontium species that I know of in the United States, and it is a Western species that we have here growing on a bunch of different conifers. It's an amazing species though, and probably extremely endangered because its hosts have basically, appropriate aged hosts have dwindled. Now, whether or not, if we leave alone the current white cedar forests that are regrowing until they become old growth white cedar forests again, whether or not this mushroom will disperse to those older forests once they reach that age class remains to be seen, but I would hope so. That would be nice. Moving south from that Atlantic white cedar sort of belt into the Gulf Coast, this is really clearly going to be some sort of bioregion. There may even be 2 or 3 mushroom bioregions nestled in here, because as we'll see, although Florida comprises part of the Gulf Coast, Florida is kind of its own thing. But anyone who's been there knows that anyways. The Gulf Coast has this fire-dependent pine savanna where there's sort of low understory fires that clear out the brush, and you get saw palmetto and things like that growing in there, as well as these amazing evergreen oaks with bromeliads that feel very tropical. And so you get all sorts of mycorrhizal diversity with the pines and oaks. Anywhere in the world that you get pines and oaks, you get mycorrhizal diversity. These are the 2 sort of tastiest trees. If you say to a mushroom lover, oh, we're going to go visit a pine oak forest today, everyone's going to want to go. That's always a good time for mushrooms. And that's what the Gulf Coast has a lot of very rich pine and oak savanna. Whoops. Here's an example of a mushroom that the United States distribution of is primarily along the Gulf Coast. This is, I wish I could show you the mushroom. Hold on one second. I'm going to delete this so you can see it. Macrocybes titans. It is famous for being enormous. If you have ever seen the picture of the guy holding the mushroom that looks like it's as big as his torso, that's this species. I think there are good, well-documented records of single fruit bodies weighing 45 pounds. So it's truly an incredible species. It's in Florida. It's in, Texas. It's in the Gulf Coast states, and it links the United States with Central America and Mexico and northern South America. And I like to call, from a biogeographic perspective, Florida is northern South America. Now, that's a little bit of an overstatement, but there are a few mushrooms that really do seem to link South America to the United States that are found in Florida. And this is because of the abundance of tropical hardwoods like Bursera and Conocarpus, as well as beech trees like pigeon plum, cockaloba. So here are some mushrooms. Tropical red Paxillus, Leucopaxillus gracilis has been found in northern Florida, funny enough, not southern Florida yet, as far as I know, that pretty much occurs in the Antilles, Central America, and then South America. So it's a mushroom that you really would never expect to see in the United States if it were not for the subtropical and tropical influence on Florida. You get these sort of endemic mushrooms to Florida that really respond to the sandy oak pine habitat that you don't expect to see anywhere else in the United States. Anytime you get a peninsula of any kind, there's a potential for reproductive isolation, and Florida is basically a state that is a peninsula. It's isolated on 2 sides by water, and it's got that heat, that tropical influence that make it so the biotic and abiotic variables are different there than anywhere else in the United States, and the fungal evolution has sort of followed that ecological distinctness, and you get a lot of sort of fungi that grow there, and you don't see them anywhere else. Pigeon plum. This is a plant, if I remember correctly, in the Polygonaceae, cockaloba, and it's got its own mycorrhizal chanterelle. So any mycorrhizal species with a polygonaceous plant is pretty interesting, but the fact that it's a chanterelle is even more charismatic. It's called the pigeon plum chanterelle, or the flamingo chanterelle, and it is a Caribbean mushroom, really. It likes the Yucatan Peninsula, it likes the Antilles, it likes Florida, and I suspect there's actually a montane species of cockaloba that's in central Mexico, and that's why you get these montane records of something either equivalent to or very like Cantharellus cockalobae. But this is an amazing species that really you're only going to get in the United States if you go walk along the beach in Florida somewhere. So no matter what way you slice it, Florida is going to be its own bioregion, maybe 2 or 3 different bioregions from the perspective of macrofungi, partly because of its polypores. So Gilbertson and Ryvarden did a monograph on the polypores of North America, and they said, we gave up on Florida. We're not even covering Florida because there's too much going on there. We're not going to cover it in detail. I heard some statistics somewhere, and I forget the exact percentage now, but it's like if you draw a line through Miami and only consider the southern tip of Florida, there's some double-digit percentage of American polypores that are only known from the area below that line. It's really an astonishing assemblage of polypores, partly because of the diversity of woods there. There are all sorts of genera that you don't expect. You don't hear these names for polypores elsewhere in the United States. Abundisporus, Ceroporiopsis earlyella, Navispora, Rhytoporia are things that, I certainly never think about in California. You can barely tell the size of it on the upper right there, but that's sort of a pinky nail sized polypore growing on the fibers of a sabal palm. This Fuscoporia calomorpha on the upper left is probably the most common polypore in southern Florida anyways. Tomophagus is that yellow elephant foot polypore on the bottom left there. All sorts of distinctive polypores in Florida that lend it some of its flavor. So you could really make a lot of arguments for Florida having multiple bioregions, important bioregions for American macrofungi. Here's one, Pulchroboletus rubricitrinus. My animations aren't coming through very well here, but oh, here we go. This one has a puzzling distribution. Now, I really don't know boletes well enough to know if this is a coherent taxonomic unit or if there's multiple species going on here, but you can see that it loves Florida and then it sort of loves central eastern Texas, and there are ones and twosies in Arizona and California and the Northeast, and I really don't know what to make of all that. I suspect that there's probably more taxonomic confusion going on here than actual distributional oddity, but the fact that Texas and Florida seem to have 2 core chunks of this mushroom's range is interesting. There's something to be thought about and chewed on and investigated going on here.
Can it be live oak? Not your live oak, but our live oak?
Probably. I don't know the distributions of oaks in the East at all. So if you're saying that there is some oak that is sort of primarily distributed in Texas and Florida, maybe, but is it missing from most of Louisiana and Georgia? Or are there just not as many observers there? You always have to remember that these maps are heavily biased towards where there are concentrations of people using our naturalist, and that some states have more and some states have less. California has a huge abundance of observers, so it sometimes looks overrepresented in species distributions, whereas Louisiana has relatively fewer per capita, so we do have to take that into account.
But look at the thing I just posted. Sorry, not to be whatever, but look at the map I just posted in the chat.
So what is that?
That's our live oak.
Okay, so maybe it actually is missing from most of Louisiana. It's actually only the southern toe.
Quercus virginiana, and if you see up in, I only am curious because I found it in Virginia. I'm one of those bad dots, but it was under live oak.
I love this because you knew enough about the trees to have a clue or an inkling or sort of spidey sense that it might be associated with this tree, and that map looks like by itself could explain a fairly large chunk of the variation. So I do think that this is really what it takes, is basically people who are knowledgeable about at least the broad strokes flora of their area, just staring at a few species maps, idly looking at them, and coming up with explanations and then testing them. Say, does that really hold? Or what additional variables or factors do I need to add to that explanation to really capture the species? Jack, is there a geological or ecological history which explains the numerous anomalous species which are found in the Sky Islands? I mean, the ecological history that I mentioned earlier is that I think that the oaks were once a contiguous belt across the eastern United States and into Arizona, and into the Sierra Madre of Mexico, and that as climate changed, that band became thinner and thinner across Texas and New Mexico and became more arid, switched over to pinyon juniper forest in a lot of those areas. And so you get remnants, basically relictual species in the Sky Islands that have now actually, in some cases, we've got evidence that they've diverged. They're genetically distinct from their eastern relatives that they presumably evolved from.
Yeah,
But
that actually speaks to another question that was actually in the chat. Would you expect some of the oak-loving species in the Sky Islands to speciate and separate, and could it have already happened? So it sounds like the answer to that is yes.
yeah, we actually have examples of both things occurring. Some of them appear to be genetically quite similar to their similar species from the east. They don't appear to have diverged very much yet, whereas other ones appear to be significantly different, both in genetics and to some degree in morphology on some of these.
Thank you. I missed that.
Okay, yeah. I also showed a little slide if you watch the recording later of some of the papers that I tried to read to inform myself about whether or not that hypothesis holds any water, and I am not totally convinced by my own explanation yet. I do want to talk to people who are more familiar with sort of paleoecology about when that fragmentation of oak continuity happened, or if I'm overreading that that continuity as sort of an explanation for this phenomenon. I'm not sure I've quite got my finger on it. Adele said, have you developed or put together any maps of the historic ranges of oaks? No, so it turns out that the mapping historic ranges of trees is hard for a lot of reasons. They don't necessarily always fossilize in an identifiable way, but you can get broad strokes ideas of where genus level distributions were. And some of the papers that I had on that slide a little ways back were actually studies attempting to do that, reconstructing the paleo distributions of some of these like pines and oaks and things like that. Okay, moving on here. So I'm briefly going to mention Hawaii. I know Jack has been doing a lot of work there recently, and there is sort of a reinvigoration of people's interest in fungi of Hawaii, and I think that's extremely important. Because, and this actually brings up something I should have mentioned earlier. The reason that this talk is themed around the United States is not because I am the most patriotic person in the whole world. It's because it's a discrete boundary that gives me something to chew on. If I focus a little bit arbitrarily on the country that I happen to have a passport for, then it makes it more doable for me to think about. I can't focus on biogeography of the whole world right off the bat. I need to start locally and go outwards from California. But you end up with these really ridiculous situations like Hawaii is part of the United States, even though from a biogeographic perspective, it's part of a totally different realm. So there are these things that are fairly well established called biogeographic realms that are kind of global in size. They're big enough to encompass whole continents. And Hawaii is really a lot more like I don't know, Oceania, Australia, New Zealand. It really doesn't have that much in common with the contiguous 48. It doesn't really make sense to consider it part of the United States, politically, socially, culturally, or ecologically, and yet it happens to be one of the United States, so I'm going to mention it. Hawaii has a lot of threatened biodiversity, up to and including the fungi. I mean, if you paid any attention to the news recently, you know that climate change is dramatically affecting what the future is going to look like for Hawaii. But that has been underway for decades because of invasive species. Invasive species are the #1 threat to Hawaiian native biodiversity, whether it's birds or plants or fungi. And I suspect that the diversity of things like waxcaps and stinkhorns is equally threatened. Hawaii has a lot, especially its higher montane forests, a lot of diversity that, because it's so remote, Hawaii is a very disjunct island. It's surrounded by, hundreds or thousands of miles of ocean on all sides. You don't expect the mushrooms there to be exchanging genes with mainland populations with any frequency. And so you expect a lot of divergence and vicariant species to emerge there. Jack can probably talk more about this at the end of the talk if he has more to add about it. But this is a place that if you go on vacation in Hawaii, take some time to try and get into these native habitats and document what fungi are there, because I don't think we're going to have a window to get to know them forever. I think they are probably going to be critically endangered sooner than later. So I want to leave you with the sense that we're still putting these bioregions together as a community. We are going to have to work on this for years to ever have a sense of some sort of coherent and stable idea of where major mushroom bioregions might exist in North America. But I've given you some leads, some starters for how to think about it and how to pursue it. One of the most fun ways to do this is to choose an area that you know well, or to choose a species that you know well, and just go on iNaturalist and just stare at the map for a while and say, does this make sense to me? Does it correlate with anything that I understand? Or is there a certain set of variables that I could become familiar enough with to explain why this mushroom is where it is? And over time, as a community, as a set of mycological associations that talk to each other, I would love for there to be sort of a workshop or a day at these annual NAMA forays or the annual MSA forays where we say, okay, what progress have we made on sort of characterizing, establishing, and publicizing what the major mushroom bioregions are of the United States? So you have a role to play in DC. You're right in the middle of a sort of conjunction of the meeting of bioregions in the Eastern US. So you've certainly got a lot on your plate to think about out there. Okay, I'll quit this for now. We can take questions or just talk for a while.
Oh, that was awesome. Thank you so much, Christian. I know I got a lot of private messages during that too about just how stunning your photos are and what a terrific talk that was. So really very cool. I did pull out one question that I saw a bit earlier, but it sort of jives with what you were just saying about Hawaii and the recent fire there. What kind of changes would you expect to find in many of the Canadian forests with all the forest fires? And I guess just to add on to that idea, I mean, obviously some level of fire is necessary and important, but it seems like we're creating bigger fires by having fewer. So I don't know, do you have thoughts on that?
I don't know what is going. I'm not familiar with Canadian forests. I've spent almost no time in Canada. But from what I understand about the fires going on up there, they are really, really intense fires, potentially the kinds of things that we call type conversion fires, where a habitat goes from one kind to another. It doesn't survive the fire as the same habitat. It doesn't just regrow as coniferous forest. And this has happened in California, places where mixed conifer forest burns at such an intensity that it does not regenerate as conifer forest. It may become permanently converted to a different type of forest, or even shrubland, chaparral, or some sort of hardwood, because the climate is also changing simultaneous to the disturbance of the fire. So you don't necessarily expect to have the context that forest once grew into after the fire. So I would expect that it's going to have a net negative effect for all of the mushrooms that are evolved for living in those boreal forests. And it's not clear to me what we should expect to see growing up in its place. Certainly not from a fungal perspective. We don't know very much about, the extent and intensity of these fires are not something that we're used to. We don't really know. We're running the experiment, and we're going to find out to some degree for the first time what it looks like. What are the consequences when you shift the climate on a global scale and then perturb it with things like forest fires? So I don't know. I don't expect it will be a net positive for fungal diversity. I think the Kennedy lab, I'm not remembering who it was. There was a paper published recently on the sort of projected trajectory for the diversity of ectomycorrhizal fungi in Pinaceae forests of North America. And the basic summary was that the outlook doesn't look great. That diversity of that assemblage of fungi is expected to decline in the coming decades because of climate change.
Thanks. No, that's great. Well, if anyone wants to just hop on and ask a question, you don't need to ask it in the chat. But I will read one that's in the chat now. Could you talk a little more about your work with IUCN?
Yeah, so let me just actually answer a few of the other questions that I saw here. Johnny B said, is the concept of an ecoregion useful to your analysis and research? Kind of. It helps me focus, in a sense. The reason we called Mushrooms of the Redwood Coast our first book, the reason we called it that is because it was a natural bioregion. We didn't really want to follow a political boundary because we didn't find it very meaningful. Like we didn't want to write mushrooms of California. We wanted to write something that the mushrooms themselves, so to speak, recognized, something that was meaningful to the organisms that we were looking at. And I always try and couch my research in some sort of ecologically bounded way rather than a politically bounded way, even though this talk I said tonight was United States. That's really not my preferred way of operating. So it is useful. It's not incredibly useful to analysis, but it does help to structure my thinking and give me a scaffold for thinking about deep time and deep ecology. Adolf mentioned that there are snowmelt fungi in New Zealand, which is an excellent asterisk that I'm glad you pointed that out. Thank you, Adolf. And yes, Kathy Cripps has a famous article on snowbank mushrooms, but I don't think it includes an exhaustive list of the snowbank species that is, I think, sincerely, deeply overdue. Elizabeth Hargrave mentions that, yes, there are birds that you find in Southeast Arizona that sort of are Sky Island specialties, although if you go into Mexico, they're all over the place in the Sierra Madre. That's something that we always have to remember. Just because they're restricted to a very small area of the United States does not mean they're restricted to a small area, because they may have tons and tons of suitable habitat in northern Mexico. Okay, so going back to the question about the IUCN, I haven't done an IUCN workshop in quite a while. They sort of took a hiatus over COVID. But basically what happens at the IUCN meetings where we focus on macrofungi is that a bunch of people who are familiar with macrofungi in North America or wherever the meeting is being held get together, and we take a list of species of mushrooms that we think might be rare or endangered, and then we do what's called an assessment. And it's actually a very concrete set of steps that you are asked to walk through, where you assemble data about where that mushroom has been found, what kinds of habitat associations it's had, what risks or threats there are to its population in the coming years. And then you make a recommendation for a ranking. So the IUCN has a number of different rankings ranging all the way from critically endangered to least concern, which is, you know, most endangered on one end to not very endangered or really thriving on the other end. And basically make a recommendation about what each mushroom might be. A lot of them, unfortunately, end up being not enough data. We don't have enough data to say. That is the most common outcome of an assessment is data deficient. We don't have enough data to run it through the sort of assessment process. We have been successful in ranking a few mushrooms as rare or endangered based on their close association with a particular plant that has more data associated with it. So if you get these one-to-one correlations between the distribution of a mushroom and a plant, you can sort of piggyback off of the conservation assessment for the plant. So that's one way we've been able to do it. But it is useful. It doesn't carry any legal weight. No one is obligated to obey any sort of behavioral change or any sort of, there's no policy recommendation that comes with an IUCN ranking. So it is up to local land managers and federal and state institutions to take that data and do something with it and to implement plans for conservation on a local scale. Unfortunately, the longer I spent at IUCN, the more I realized we were kind of spinning our gears. Like, conservation at this point in human history has to do with changing our entire civilization. It's not about protecting individual species. You can't really protect a mushroom and expect it to survive when its habitat is experiencing an onslaught of abiotic and biotic changes. Invasive species and climate change are far too big a challenge to reasonably manage. You can't do that as a land manager. Those are things outside your scale of control and influence. So I have started to really pivot towards human cultural change as the area that I recommend students to go into if they are interested in actual conservation. It's all going to come back to changing the ways that we sort of incentivize consumption and normalize consumption. The fact that we still live in a world where private jets are a thing means that all of our sort of species-focused conservation efforts seem a little bit silly. You cannot take a Tylenol to cure a chronic illness. It is going to be a sea change in how we behave as a species if we're going to conserve species. That said, there are mushrooms with ranges that are small enough that keeping invasive species at bay probably does buy a couple decades of time for that mushroom to continue to survive. So I'm not saying that there is no immediate conservation work that can be done to help mushroom diversity. For example, that lepiota that grows under cypresses, the yellow-gilled lepiota, it has such a small known range that if an invasive plant started to sort of, like, for example, English ivy grows in a lot of those cypress stands, if it started to smother the understory in that cypress grove, it would pose an existential threat to that mushroom and would argue for immediate direct weed abatement as a conservation measure. So I'm not saying there's no place for it. I'm just saying in general, I have started to take less of a species focused approach to conservation and more of a habitat and cultural approach to conservation.
And is that because the invasive species are directly smothering at the mushroom, or because they're affecting the host? Or both?
It can be all sorts of things. So there are pathogens that kill plants that are the only hosts for a mushroom. And if that pathogen is an invasive, then it's a sort of second order effect. Sometimes, like you say, it's habitat quality modification. Like if English ivy grows into a cypress forest, it's not that it's killing the cypress. The cypress actually probably does fine, but it changes the nature of the understory. It changes the quality of the duff. It changes the physical texture, changes light penetration. It changes water penetration into the soil. So it's not like it directly kills the mushroom. It just changes its habitat to the degree that the mushroom probably can't continue to make a living.
Yeah, some of the patches where our club used to go hunting for morels have been completely taken over by Japanese stiltgrass or Japanese barberry. And it's hard to tell whether we just can't see the morels anymore, but it seems like they're not coming up in there anymore.
Yeah, I have heard the argument that, you just can't see the mushrooms under the English ivy, and I don't buy it at all. I think the fact that you can't see them is because the understory has changed in its physical qualities to such a degree that the mushrooms have also noticed. It's not just your eyes. Your eyes are telling you an important ecological fact. Our senses are actually quite well set up to be sensitive to ecology. It's just that we've lost the cultural sort of empowerment to talk about that in a subtle way. We don't have the sort of lived experience of depending on ecology the way our fairly recent ancestors did. So I think you should trust your senses. That absence is not simply a trick. You're noticing something quite important.
You have another question from Adele, who was curious about what percentage of your research occurs in the field, and what are you documenting in terms of the habitat eco-community?
I try and make all of my research happen in the field, which means that you never see me publish anything anymore because I hate sitting in front of a computer. If I'm lucky, I won't do any more. But one of the things that I'm working on sort of most actively right now is mycoflora of the Channel Islands. So it's an archipelago of islands off the California coast. And basically we've done quite a lot of field work out there. I started in 2012 doing sort of occasional visits. It's sort of hard to get out to islands, sort of by the nature of them being islands. It's not, you can't just drive out there. You have to arrange a boat out. And it's sometimes the weather prevents you from getting out there. But we've done quite a bit of field work out there. And now we're sort of trying to piece together the story of which mushrooms are associated with island endemic plants, which mushrooms may be archipelago endemics. We don't really have bulletproof evidence that there are Channel Islands endemic mushrooms yet, but we're pretty sure there are. We just need to prove it now. So we've done a lot of field work, but on the back end of the field work, there always comes a lot of digestion and summarizing and analysis that I'm trying I'm actually taking a year off from teaching at UC Santa Cruz this year to work on some of my outstanding projects that I have never given enough time to actually finishing. So I'm taking a poor man's sabbatical this year.
Yeah, I feel like we could have a whole nother talk on how do you solve that kind of mystery, to tell if something is endemic, or what, but.
Yeah, part of the problem is that you can say that there's a Channel Islands endemic, and it'll hold true for 10 years, and then someone on the mainland will just find the population of mainland, and you're like, oh no, it's not an endemic at all. We just didn't know where to look on the mainland, and here it is. And that actually happened. We found this in January. We were out on Santa Cruz Island, and it was one of the wettest periods I've ever seen on Santa Cruz Island. It was incredible. And we were finding a little purple Clytocella, which is an entomomatoid mushroom, under a Channel Islands endemic buckwheat. And we thought, oh, this is for sure going to be an endemic species because it's growing, you know, on the Channel Islands, under a Channel Island endemic plant in a very harsh habitat that only gets wet every so often. And then I went on to, you know, iNaturalist and found out that Jacob Polk had found it on the mainland, you know, a few years earlier. So there you have it. Yeah. So it everything is endemic somewhere, and the boundaries
although
of that
it could go in the other direction, I guess. I mean,
certainly, yeah,
bi-directional. So maybe, you know, maybe it was endemic until you went to look and see if it was endemic. It's like yeah, the closer you get to your subject, the more you change it.
Yeah, definitely. I mean, that is another difficult thing with mushrooms, that it's never possible to rule out human introduced, you know, human introductions of spores or plants, especially in a place like Santa Cruz Island, where there are a lot of international visitors and national, you know, a lot of ferry traffic from the mainland. People carry soil on their shoes. There are even plants transplanted out there intentionally, at least in the past. So we kind of lost the window to document a baseline for truly what was on the islands before human modification, but we can sort of make informed guesses based on what's common and what's rare. For example, there's only one record of Amanita phalloides in the Channel Islands, even though it's extremely common on the mainland right across the channel. So there are some, there is a barrier to dispersal that appears to remain somewhat distinct, even though there's a lot of human traffic back and forth.
Well, speaking of human kind of changes, there's a question about the loss of fungal species associated with the American chestnut. This is something we talked about also in our meeting in August. You know, we were talking about fungal pandemics. And yeah, so what do you, you know, when you think about a tree that, you know, was like incredibly prevalent in a zone and the kind of mushrooms that went with it, do you know of specific species that we've lost?
Yeah, so unfortunately, I don't, because at the time that the American chestnut had its total collapse, people weren't as interested in mushrooms as broadly as we are now. And we lost our chance to see, you know, what really was a chestnut endemic mushroom. And there's zero doubt in my mind that there were American chestnut endemic mushrooms, and probably lots of them, because it was a keystone forest tree. There were, I mean, I didn't realize how many chestnuts there were until I read about the history of them 2 years ago when I was visiting the Northeast. Like, it really was a profound landscape change across the entire eastern seaboard. So we must have lost, not, I mean, keep in mind, a chestnut is part of the Fagaceae, and that family is known for being excellent mycorrhizal hosts. So there undoubtedly were mycorrhizal plant or fungi associated with it that experienced a total loss of their host. And you also tend to get, anytime there's a plant that is that abundant, you get non-mycorrhizal things, decomposer mushrooms, that are substrate specific specialists. So we probably lost wood decomposers that had specialized on chestnut. There's no doubt in my mind that that was a major loss for fungal biodiversity as well, but we probably will never know. I mean, I don't know how we could reconstruct what fungal diversity was lost.
I mean, it really kind of, oh, go ahead.
Follow up to that question. If the plans hold true, they're planning a mass reintroduction of genetically modified chestnut.
Yeah.
Do you have any thoughts on doing this? I mean, that extends to other things too. I hear they're working on cold hardy eucalyptus, et cetera, et cetera. Which, do you have a general pro or con against reintroducing in mass
No,
trees?
I really don't have a reflexive position on it. I can see the pros and I can see the cons. I'm certainly not dogmatic about it. I think it's context dependent, and just because you do it with one species doesn't necessarily mean you should do it with another. I guess one way that you could find out what happened with chestnut fungi is if they succeed in doing this mass reintroduction and you actually get healthy, mature, quasi-American, quasi-hybrid resistant chestnuts in 100 years' time, and you suddenly see all these new mushrooms that had somehow survived in the spore bank for that many years suddenly appearing, that would give you a sense that there actually were these chestnut endemics that were just waiting for their host to reemerge. I don't think that's going to happen. I think that there's probably enough of a sharp cutoff between the extinction of American chestnut and the reintroduction of a not quite genetically equivalent strain that probably will act as a sufficient bottleneck or filter that we're not going to see the, bloom of the chestnut associated mycoflora again in the future. But that would be a hypothetical way to detect it.
And you have to wonder how the reintroduction will work if the fungi that the trees used to rely on for some nutrients aren't in the soil anymore.
Yeah, I guess it would probably just use generalist Fugaceae associates, so oak associates that can also pair with chestnuts. But you're right, maybe they are going to be missing their most intimate co-evolved partners, and that maybe the reintroduction won't go as well as it would have if some of those fungi were still around.
Yeah, one thing to keep in mind, though, is that there are numerous sprouts of chestnut. For example, the American Chestnut Foundation estimated just a few years ago in Virginia, there are over 68,000 of these sprouts. I've seen many of them in the Shenandoahs, but also up in Pennsylvania as well. So presumably those sprouts, I've seen them 18, 20 feet tall, and on rare occasions they do fruit. So I'm assuming that they've maintained some level of mycorrhizal association, although I can't verify that. I worked on the risk assessment for the American chestnut when I was with EPA, and they looked at the potential effects of the oxalate oxidase gene introduced into the chestnut against various fungi, because obviously you're trying to mitigate Cryphonectria parasitica. But that's a necrotrophic fungus, and the way it works with the oxalate oxidases is not a direct attack on the fungus per se. It's an attack on the oxalic acid that allows the necrosis to occur, and then the fungus moves in. So I think a lot of what I've seen over the last few years, people getting particularly wrapped around the axle about how this is a slippery slope and we're going to have genetically engineered trees, really doesn't ring true in this case, at least, because it's probably the most innocuous gene you could have put in there. I mean, it's found in dozens of grasses like barley, wheat, things we eat every day, apples, etc. So I don't think it's going to impact the mycorrhizal association that chestnut would have. But the other question you raise is a very good one. How is this going to be actually logistically programmed through whatever National Park Service, U.S. Forest Service, state folks, state foresters, to actually distribute this tree to the right places where it might in fact actually have a chance of restoration.
Yeah, it's curious.
I saw one fruiting this summer up in Shenandoah, so that was pretty special to see.
That's cool. Yeah, it is. I mean, that's something I also learned when I was visiting 2 summers ago, is that there are these sprouts, these sort of like, it wasn't a full disappearance. There are some sort of sucker sprouts, or I don't know exactly what their biology is, but I sort of doubt that they would maintain the full suite of mycorrhizal associates, because we know, for example, that there's a very big difference in the assemblage of mycorrhizal fungi with young pines versus old pines, even of the same species. And if you metabolically change the host, you also change the kinds of fungi that host can support. So it's important to sort of try and put your perception in the space that a fungus is occupying, which is that even 2 trees of the same species can be quite different, because you as a fungus are so intimately dependent on their physiology and the whole context of the ecology that they provide to you, they're not going to be the same as a bunch of mature trees growing with some sort of landscape dominant cohesion with all the network effects that brings with it.
Totally, like what you said about Echinodontium balui, or I'm probably mispronouncing it, but I read that article about Bill Neal, like searching for it, and they went through so many cedar swamps, ones with very tall trees, to try to find it before they could find one that was mature enough that it actually had it. And, I think that's like a perfect example. When you look at those photos of chestnuts, they're bigger than a truck and it must be very different as a host compared to the shrimps, the kind of shrimpy.
It would
seedling.
be like you deciding whether or not to stay in a hostel or live in a mansion.
Yeah, right. That's such a beautiful way to put it in our own sad terms. Totally. Yeah. Well, I know we're going late on time, and I don't, I would feel like we could ask you more questions. But you're getting some hellos and many big shout outs in the chat. I guess if anyone has a burning question they really want to ask, then now is your chance. But mostly I would just say thank you so much, Christian. This was
thanks for having me.
Really terrific. And I just want to say, I love the message that you were giving in terms of Hawaii. If you happen to be on vacation there, document what you see. I think we could say that with where we live as well. There's such an incredible development going on, even in a very overdeveloped place. Yeah. And maybe we all need to be bearing witness to what's here now.
It certainly won't none of the landscape will look this way in another 150 years. So I do think that there is, it's going to be an interesting thing to look back upon and say, what was it like then? What value that has is a different question. Whether, I don't simply want to be a documentarian of change. I do also feel some responsibility to do practical stewardship, to sort of make space for things to get smuggled through this great spasm that we're going to be headed into ecologically. And a lot of my advice now to my students is to figure out some way to feel like you are helping a few more scraps of that biodiversity get through this great change that's coming.
That is a great message for us to end on. Oh, Jack, did you want to? Yeah, go ahead, please.
Just one small thing. If you do plan on going to Hawaii and naturalizing, keep in mind there are fungal pathogens that can be spread by humans going through forests. And I'm putting a link in the chat. If you're going from a forest that's known to have what is called rapid ohia death, sterilize your boots and all of your equipment before you go to another forest, because there's evidence showing that mammals can help spread these pathogens around. So just be mindful with your documentation.
Yeah, I mean, I do feel like it's not an overreach to say you can also consider not going. I have really started to rethink what it means for me to travel. And I've gotten much more nervous about how normalized it's become to fly anywhere. It is a kind of a remarkable derangement of human consumption that we have just gotten used to the idea that we can go across the country immediately. I'm not saying don't. I'm sure I will do it again. But I feel a lot different about it than I did 5 years ago. And things like what Jack just mentioned are an excellent example of the kinds of disruptions that happen with just a moment of incaution or ignorance on our end that have profound, existential threats to whole species and ecosystems that really argue for the precautionary principle when you're traveling, is just be really careful and potentially just say, out of an abundance of caution, I'm not going into this highly threatened area. There are parts of the North Island in New Zealand that are totally closed to visitation now for a similar reason. The kauri, which is also a myrtaceous tree, has a pathogen that has been affecting it, and because it's a sacred tree to the Maori people, they're not really letting anyone go in there. So it's true everywhere. I mean, there are places, Port Orford cedar has seasonal closures in the Pacific Northwest where you can't drive through that area because the mud on your tires can introduce Phytophthora. Humans are a major vector of pathogens throughout the world. It is one of the ways in which invasive species and microbes get moved and end up having really sincerely out of scale effects on the biodiversity that we visit.
Well, I think this is something that we can think of too, you know, on a club level about our forays, and I know we've talked about it a lot to think about what kind of impact we're having. So anyway, thank you. This was a wonderful talk and a lot of food for thought, and really enjoyed it, and got a lot of people hanging on.
Thanks
you
to
again.
everyone for staying, and yeah, thanks for having me here tonight, Annie.
And hopefully when you do come back to the East Coast, you'll let us know, because we would
Absolutely.
sure love to have you here. It is, as you said, we're in a weird location. It's like a crossroads between North and South, and there's some cool stuff, and we would love for you to come hang out with us and see it. So
I would love to. I'm sure I will.
Okay, awesome.
Good
Take
to
care.
see
Thank
you
you
all.
so much.
Bye, everybody.
Thanks, Christian. Thanks, everyone. Good night.
Bye.